Very good evening to one and all here today with us. And as you can see that we are all set to go and to start this today's exclusive session, that is a book review session on a book named as COVID-19 Detection Strategies Swab to Saliva. So we have our pioneers with us, and today we will listen that what is their opinion about the book that we have worked on, and we have tried to bring the best as possible from our best capabilities. So first of all, I welcome Dr. Paul Desmond-Sloy, who is the CEO of Oasis Diagnostics from USA, and Dr. Sriram Raghavan, who is the Senior Vice President of Janam Private Limited, Coimbatore, India. Welcome, sir. Welcome, Dr. Paul, and thank you so much for joining today. And we also have Dr. Rujita Patil with us, who is the assistant editor of Genomedan magazine run through our organization. Welcome, Rujita. And we are all from different parts of the world sitting together now, and I think this is amazing. So straight away, I think I will just pull the program, and we also have our other guests joining in soon. And to start with all your permission, Dr. Paul, how are you? First of all, I would like to know that, and then we'll go into our session.
So I am doing very well, thank you. My name is Paul Slowy. I'm the CEO of Oasis Diagnostics, and Dr. Gagi Roy Goswami and I have collaborated for quite a while. And this project came about very inspirationally, and I guess without any specific target at the time, but kind of morphed from what we believe to be a very good manuscript that would catalog the events that happened from the beginning of 2019, perhaps till the end of 2020, as far as the COVID-19 epidemic was concerned. And the title of the book, Swab to Saliva, basically describes the process that happened that put saliva on the map. So that would be a introduction. Dr. Gagi, would you like me to explain a little bit more? But that's literally how it came about. And the saliva has now become a fantastic tool for COVID-19 testing, and many companies are in this area. Would you like me to explain more?
Please, because
Yeah.
I
Okay.
think
So
from you as
I think
always.
what we tried to do, and correct me if I'm wrong, was basically catalog the events that happened from the start of the epidemic, where if you look back to September 2019 or December 2019, when I actually came to China with COVID-19 myself, there was the predominant method for testing for COVID-19 used either a nasopharyngeal swab, an oropharyngeal swab, or much later on nasal swabs. But saliva, I think there were, if I remember rightly, in December 2019, there were only 2 publications that actually supported the use of saliva samples for COVID-19 testing. And you have to move to about March 2020, when there was a global shortage of swab materials. The two major companies at the time, I think, were Copan in Italy and Puritan Swabs in the United States, and then many Chinese manufacturers. But I recall talking to the head of Thermo Fisher, which is I think it's a $25 billion company, probably one of the largest diagnostics companies in the world. And at that time explained that there was a worldwide shortage. They had a back order of something like 37 million swabs for their customers. And, if you multiply that by the many other companies that were in that business, nobody could get swabs, so the price went up, the demand went up, the cost went skyrocketing, and then a lot of researchers suddenly, it kind of dawned on them that saliva, and other specimens were also useful to be used for COVID-19 testing, and that kind of spurned a heck of a lot of activity in the saliva world. And we as a company provide saliva collection tools, and it became a very interesting time for us, because many companies would come to us and say, we need a saliva collection tool. What do we use? How do we use it? Can we use your devices to partner with our test kits? That kind of coincided with I would call it a project in the United States, which is called Rydex. And Rydex stands for the Rapid Acceleration of Diagnostics Program. And this was implemented by the National Institute of Health and President Trump to literally give, I believe it was $1 billion. I could be wrong. It might be $500 million, but literally to support the development of COVID-19 tests in general, not specifically saliva tests, swab tests, urine tests if necessary. So, any bodily fluid that could be used for COVID-19 testing. But the aim was to get as many tests on the market as soon as possible to test as many Americans as possible. And that did spur a lot of money went to companies that were allowed to develop tests, set up the manufacturing capability to develop these tests, to manufacture these tests, and then to bring them to commercialization. And I think what we see in kind of the fruit of that harvest, if you like, now. And but of that number of companies, a lot of them kind of got an interest in saliva very, very quickly. So you find that it's not 50-50 by any means, but maybe 10%, just as an estimate, of the companies that are out there would develop a test based on a swab system, but then would reflex to also a saliva sample. And the papers are right. In the beginning, it was kind of the jury was out on whether saliva was a good matrix for COVID-19 testing. But I think if you now look at the body of literature that's out there, there are many publications. I would say maybe 500, maybe 1,000 publications. And if you go back to December 2019, when there were only 2, that just literally shows the ability of saliva. And throughout the time from around about April 2020, as we mentioned in the book, all the way through to now, there are multiple tests getting FDA, what they call emergency use authorizations in the US, also getting CE marks in Europe, and then getting approvals in other countries around the world. So it's become not just another specimen. It's become kind of the mainstay of of COVID-19 testing. So it's an exciting time, and it's continuing. More and more companies are jumping into this market. COVID-19 is not going to go away very quickly, as we all know. So, the more and more tests, and I think what companies are now doing is looking beyond COVID-19 to look for maybe COVID-19 plus flu A, flu B, strep pneumonia, and various other diseases in saliva, which they can do either by multiplex PCR tests that are already on the market and using a saliva specimen too. So that's basically the background, and I think our book kind of goes into the different technologies that can be used with saliva. In my opinion, it's what I call an agnostic specimen. It'll work for antigen testing, it'll work for antibody, it works for real-time PCR, CRISPR, LAMP, you name it. And there are publications out there supporting that. So it's a very interesting time to be living right now. I mean, it's not the best time in our history, obviously, because we're restricted by the pandemic. But in terms of how saliva has contributed, I think we've done a very, very good job collectively to make that a useful specimen.
Thank you, Dr. Paul, for that amazing explanation and highlighting how saliva has become so important. It was important, but I think COVID has given a push. And right that we are into the program, and I have another special guest who has joined, Dr. Deborah Heller from Brazil. Thank you, Dr. Deborah, for joining in spite of your busy schedule. Just to go ahead with a few more questions here and there, Rujuta, would you like to show our book to everybody? I think we are very excited because I think it has been a very exciting journey through this entire project because we worked on this project from I won't call it international or global, but we really don't know how we made it because we live in on different time zones. And me, Dr. Paul, and Rojita, we are in 3 different parts of the world. And through emails and few documents sharing, we just completed this, and everything has happened online. So, Rujuta, I would just because I think only Rujuta is here, where she has the physical copy of the book with her. You and I didn't received it, so I would just tell you. I mean, you can just show the book to everybody, and that's a, I mean, amazing feeling actually.
Yes, ma'am, I do agree with it. So I was very much excited to get the book in my hand.
Great.
This is really special. So this was definitely a very exciting journey. I think we all are very much excited and equally enthusiastic about this book, and especially after seeing the hard paperback, the hard copy, and getting this in hand. This there are no words to explain what this feels actually. So very much excited, ma'am. So this is the book. It is available in Amazon to
Yes.
purchase. So please do check out and buy this book. We have given a lot of efforts, and there is an immense knowledge about right from the structure of COVID, from its history till the recent diagnostics of the saliva and how it is useful in detecting COVID-19, and the global market, as Dr. Paul has already explained in brief about all this. Everything is given in detail in this book. So please do check it out.
I think the icing on the cake regarding the saliva market and other insights wouldn't have been possible for us without Professor Paul's constant guidance and support. Thank you once again, Dr. Paul,
Thank you.
for everything that you do
No,
for us.
it was a very collaborative effort. Your efforts are sterling in India. In the fact that you promote saliva throughout India and have done from the very moment we met, and you work tirelessly to make sure that it is not a forgotten specimen. So I thank you, and I thank the team here that's put this together. And I challenge everybody to understand what saliva can do for the future, not just for COVID-19, but generally speaking, how it can help mankind in the future to be able to diagnose diseases non-invasively, quickly, and just the best way to do it.
Right, right. I agree. So I will just like to ask Shriram sir, because I think Shriram sir and Dr. Paul, they are both my mentors. And today itself, I think I was talking to Rujuta a little while ago. The direction after coming in entrepreneurship, the direction that I got, I think these are the two people who are very, very instrumental. And well, so Sriram sir, we all would love to hear from you and your views about this particular book, and then I'll come back to Dr. Paul. I think
Good. So thank you so much for inviting me. It's a pleasure, honor, delight, whatever you may call it, whatever adjectives. But truly, I'm delighted to be part of this wonderful moment. My heartfelt appreciations to you, Dr. Gargi, and to Dr. Paul and to Dr. Juta for making serious efforts to compile this book. And what an opportune timing for the launch, especially when we are looking at a new variant, another Greek alphabet, as I was mentioning before. Yes, WHO did skip one alphabet, which is called Z, for reasons which I think audience will understand. I'm not going to go in depth. Of course, I'm not sure what diagnostic test can be used for detecting that. So now we are into Omega, which is Omicron. Now, the reason why I like this effort from Dr. Paul and Dr. Gargi and others is that I'm working with Government of India for the last 18 months or so with various departments in identifying various proposals on COVID diagnostics in terms of fast tracking, especially for a country like India in terms of mass screening of population. So this is about close to 100 startups that I've been, in a way, I've been involved in some way of funding them directly or indirectly through angels, through venture capitalists, etc. This is especially in the last 12 months or so. And many of them are working on different models of diagnostics. And they're trying to improvise all the time in terms of sensitivity, selectivity, specifically looking for the variant, for biomarkers, for symptoms, etc. The main problem is that RT-PCR takes time. We all know that. So people want something which is quick and easy, especially if you're going to come by, let's say, a train station, or let's say if you're landing by bus. Of course, aeroplane is always there. When someone lands into, because India is almost like a mini European Union, it's like there are different states. Each state, health is a state subject in India. So what happens is each state can impose their own quarantine on top of what the federal government is going to impose. So that puts a lot of problem, especially for those who are on the move. And this is a serious problem because let's imagine you have to spend 7 days in quarantine. Imagine the cost of hotel stay and all of that. Now, RT-PCR itself takes a lot of time. when I say lot is a relative word. Someone who just wants to take the flight to, let's say, to Dubai or someone, very recently I was reading that it takes at least 3 to 4 hours for them to wait in the airport. And many times families have missed the flight. We have been reading this in the news. Imagine if Saliva could be a very easy option and we get quick results. It could be just a vague idea. It need not be all the way sensitive, etc. Just yes or no. As long as people have, it says there's no COVID infection, people are able to fly. That itself is a big relief. I think from that perspective, from travel perspective, this could really prove a new way. I mean, making money, getting into the startups, getting into science is one part, but practical application in terms of many different families whom I know could actually get benefited from this. Number one. Number two, many times in India, especially, at least I have been, getting a lot of queries on this. When they take aged parents or aged people to hospital, the first thing that they do is to, get a COVID test. Now, this is a nuisance. Already, let's say you are taking someone suffering from a heart attack or any other serious ailments, you want urgent treatment. But many times hospitals are, I wouldn't say heartless, but they are bound by the state rules and federal rules. They say, please get a COVID test. Now imagine if you're going in the middle of a night, because most of this disease wouldn't tell you and say when I'm going to hit you. So, it hits in the middle of the night, you take a patient to hospital. Imagine you're stuck there. And if, where are you going to get your COVID test done? Diagnostic labs are closed, samples are going to be collected next day. This is a serious problem. Again, I imagine collecting a saliva swab and then, quickly, doing this. That would be wonderful. So I had been telling all these 100 startups, to also look at saliva. Of course, now the publications are coming up, as Dr. Paul rightly pointed out. It's going to be a fantastic improvement. More so ease the pain, faster results. I'm not seriously looking at, sharp sensitivity like RT-PCR. RT-PCR could come in at the second level. This is something at the early stage diagnosis is all that I'm focusing at this point of time. If this could really have something, and I think we could push many startups in India, at least from Indian perspective, that I can say that there is a government funding available. We can actually encourage them to work on it. It's more from a public community service perspective that I think I would really want to appreciate Dr. Paul and Dr. Gargi for actually inspiring many of the startups, because I think I would look at this book as more of an inspiration because you need inspiration for people to think. Right now people are not thinking that saliva is a very important tool. I think this book should serve them and tell them, hey, look, saliva is an important one. Don't ignore it. So, spitting is a serious business. So that's all I would say. And from that perspective, continue to do the spit, but it's more, from a medical relevant perspective, and you can actually, make money, ease people's lives. And the last point that I wanted to conclude is, as Dr. Paul said, saliva is a sector agnostic, discipline agnostic, and, the disease agnostic tool. We can actually do a lot of things. There is one more interesting thing which I was reading, that many of these startups are also using, let's say, far infrared, radiation, and many of the other things for, kind of non-invasive, non-touch-based, quick, detection of symptoms, and also partly for COVID. Of course, that sounds like a magic, but eventually people are working on the technology. If we could actually have a device very similar to what happened in Israel, is that you just probably breathe into it or spit into it. There is a combination of the nasal output as well as the saliva that actually happens in an encapsulated space. That I think could also be a very good combined tool, because there could be biomarkers in both, where we could actually give a quick readout, at least to look at false negatives in terms of how things go forward. And I think Israel has also been giving a very good support. And I would also suggest Dr. Gargi and Dr. Paul that if this can also be popularized, especially the book in Israel. There are a lot of startups and many accelerators that I know is also wanting to look at this. They can actually use this as an inspiration, because in Israel, the funding is, of course, definitely more. The startup ecosystem is much more mature. So if something happens there, obviously countries like India or Brazil can actually import the technology very quickly, and they can actually scale it up, because scaling is very good in India. But the original source and original invention can actually happen from there as well. So I'm just, being very open from that perspective. So with that, I would like to conclude. Thank all of you for this wonderful effort. And thank you, Dr. Paul, one sincere word of appreciation, because I think you have been inspiring Dr. Gargi all throughout. You have been a rock solid support. I can see that you have been up at 5 a.m. in the morning for this event. Thank you so much for doing this. And of course, Dr. Gargi, you're always, fantastic source of continued inspiration in terms of what you're doing. I think you have launched a magazine with Rijuta. You've launched this book. You have been launching. And of course, a fantastic timing ahead of the SALC 22, which is going to come up, because I think you're going to use this book as a projection of great effort of what Saliva can do. If that happens, I think I would consider this whole effort as a very, very successful way to forward to inspire people in terms of what can be done. Thank you.
Thank you so much, sir, for all the nice words and appreciations that you have in basket and you have told about us. I think we have to come more, do something more to, come up to all of your expectations. So, Dr. Paul, just going to you once again. Um, how was your experience in this entire book writing project? I think that is one thing everybody would like to understand from you, because I think at least I know that I don't know about Rushuza, but I have asked many silly questions every time to you. So how did you deal with these two people in front of you, I mean, in this entire
Oh,
project?
I guess it's my first book project. I have a number of book chapters that I've written either collectively with other people or by myself. By Dr. Streckfus, Charles Streckfus asked me to write a chapter. Dr. Dave asked me to write specific chapters. So I have book chapters on my own. So I don't, but I've never really gone as far as writing a book. So this is a first experience for me as well. But if as you recall, it started life as a manuscript and just kept going after
Right.
that. So it wasn't a conscious effort in the beginning. I don't think it just happened like that, right?
Correct, correct.
One other thing I wanted to add to Sriram comments, because I think they're very valid, especially in India as well, which is kind of the I guess it's the hotbed of things that are happening right now. I think he should be his comments should be inspirational to people. But what's happening here in the U.S., which might reverberate around the world a little bit, is that President Biden gave a whole bunch of money just recently, specifically targeted what I'd call rapid point of care or home-based tests. So they've kind of de-emphasized tests like real-time PCR because they take so long, Dr. Sram. They've now focused on tests that can be used by people, either buy them in a pharmacy and take them home, or they can just buy them locally and then do the test, or they run them in a drive-through that might be some sort of point of care test. So it's kind of shifted the market a little bit to the point where the tests that are going to come to market will, some of them are capable of testing for COVID-19 in 1 minute or 5 minutes. It's really fantastic how these technologies work to be able to provide a signal or a result in just 1 or 5 minutes, and simple enough that people like you and I, or the man on the street, can actually use them, which could turn the whole epidemic around. That means we go out less, because we go to the pharmacy, we buy the test, we find out the fact that we're positive, we quarantine ourselves, and those types of things. So I think this is maybe a change in the way that COVID-19 testing is done. But once again, I think it'll inspire companies to look for specimens that are easy to deliver. It's much more easy to deliver a nasal swab or saliva rather than a nasopharyngeal or an oropharyngeal swab. So I think the market's beginning to shift again. So maybe we have to have a second book in 2 years' time that talks about the changes after this. But yeah, I mean, it was a very positive project and very, very heartening, and it was inspirational for me, and to be able to work with this accomplished team. So yeah.
Thank you,
Yeah,
Dr.
so
Paul.
just to comment to add, I think, Paul, that's a fantastic point that you brought about. In India, there are two such products where you can actually order these tests online, and you can actually,
Mhm.
they will actually send you a kit. It's a kind of an
Right.
antibody-based detection, and it's very quick. Just put a pinprick, and then they will actually tell you in about 30 minutes or so, just an hour. The main
Right.
problem comes in is that the official recognition for these results that you take, then let's say I can't take these results. I mean, I can take these results for me to decide, as you rightly pointed out, whether to stay in quarantine or not. But I
Mm-hmm.
can't take these results to an airport. When I enter, the security will kick me out. And
Right.
because he needs an actual government validated certificate for
Yes.
me, even to, it's
Right.
almost like a security. I'll become a security risk. So that's a problem.
Yeah.
No, it makes sense, and even antibody tests are being kind of de-emphasized
Yeah.
here in favor of antigen tests, which are more accurate, more specific. Or what antibody tests will tell you, whether you have sufficient immunity moving
Yeah.
forward. And I have pretty high antibody levels because I had COVID in 2019, but then I was also vaccinated with the Pfizer vaccine, double vaccinated. So I haven't actually seen what the level are, but there is a company that's actually testing my antibody levels for me, just to let me know exactly where they are. But they're basically diagnosed from the early results before I was infected, that my antibody levels were way above the levels from a normal individual, and that I'd had a mild to moderate COVID back in November 2019. So, but these tests, a lot of the tests work, and as I think as Sriram mentioned, sometimes speed is more important than accuracy. So maybe we can sacrifice it. Maybe it doesn't need to be 100%, 100%. If it's 90%, sensitive and 100% specific, and we monitor regularly, then I think we're going to catch what we need to catch.
Absolutely. So I think in this context, I think I would
just got Dr. Gari, just a point to add. I think
Yes.
nature is, as Dr. Paul was saying, I think nature has selected Dr. Paul specifically for this book project because of his high antibody levels. Maybe.
Yes, maybe. Maybe. And so I think in our book we have tried this only because what I found that there has been information which are coming out in different aspects, like the people talk about serological tests, antibody tests, and also different types of advanced technology. So from that point it came to our mind that we should bring all these things together in one place. So I think, Rujuta, I would like to tell, I mean, just can you highlight some of the concepts that we have put through, and it is very presented in a very very lucid language, because we thought that this should reach even to people who are not from the scientific background. Yes, I agree that there are many scientific terms or terminologies used, but I think it is so so simple English that if you can read English you can understand this book. So I think, Rujuta, if you can highlight like what are the kind of tests, or what are the detection methods we highlighted, maybe just a very brief summary. Yeah.
Yes, ma'am. Like we have, as I this like we are talking about this book now. We have started right from the history, the structure, how pathogenesis, what happens once virus enters the body. Like when I'm saying what happens when virus enters the body, this is as simple as that. It's been explained in this simpler manner what actually happens. Later, once it has entered the body, how can we detect? So we have been listening about RT-PCR, the CRISPR, LAMP, RT-LAMP, all these technologies using blood as a sample. But surprisingly, all this are effectively used by using saliva sample as well. So this is the specific, the key or the important factor what we have highlighted in our book. And it is not just that we have told that this is possible. We have also explained the principle behind each of this test, how it is explained, both pictorial representation as well as in the written format. All this has been explained. And after this, we have gone towards what saliva actually how it has made its place in the market, and how the global market of saliva and the detection of COVID-19 is growing. What was the past status, and what is the present, and what will be the future? So everything has been given in detail. What are the advantages of using saliva samples? Like even comparing it with the nasopharyngeal swab, how advantageous it can be. Like few of the examples, like in the mass and community screening, even in the neonates, it can be used with the elderly patients. There are so many advantages which saliva has got. So all of these has been explained. Also the detecting tools, the saliva collection tools have also been put up here. So if somebody wants to know about that and get a brief idea, everything has been properly put up in a tabular format as well. So it is very easy and self-explanatory. After this, an entire thing has been packed into this mini book, I have to say. And we have tried to execute it as much as possible to make it simpler for the people to understand, and also for the upcoming researchers to take up saliva as a research topic and move in this direction. Like we are trying to inspire the young researchers and also the established researchers to look into saliva and continue the research in this aspect as well. So yes.
Thank you, Rujuta, for that. And I would
So
just go.
just a point to add, Rijuta, it's a very well said point. Just a comment on that. I was telling Dr. Gagi that because of COVID, one profession that actually took one of the maximum hit in terms of the income is dentist, because most of the dentists, they had to shut down their clinics, because what do? You have to open the mouth first, right? When someone is wearing a mask, you
Yes,
can't do that,
yes.
correct? So
Yes.
dentists suffered a lot, at least I can see from Indian perspective. And I think many of the dentists, they really wanted to have a fundamental understanding of the diagnostic procedures, because if they have to come for a dental procedure, they have to first, actually, send them for a quick COVID test. And they are not very sure about, what is the principle that is going on, how do you rely on some of this test, etc. And I think this is a very opportune timing. And more importantly, at Salsi, I think you should insist why it's important for dentists, especially for them to restart their business.
Yes,
Right,
yes.
sir. Right.
So
So,
we have a dentist on the
yes,
line. Dr. Deborah Heller is practicing
absolutely.
dentist, and she probably has said more than anybody else through this epidemic, right?
Exactly, exactly, Dr. Paul. I was just going to say that like that you have to look beyond your profession. So here is a living legend, living example, Dr. Deborah Heller from Brazil, and I think Dr. Deborah is inside another meeting, but still she is here. So I just caught her once again. So Deborah, thank you for joining, and I'll just straight away ask you that how what is your opinion about our book, and definitely as a scientist yourself and a practicing dentist. So to all the dentists out there who are watching, who will be watching this, so dentists do wonders, and with saliva, we're the people who deal with it day in and day out. So actually you can see it as a friend, not as a foe. Dr. Deborah, I would just like to ask you about your opinion regarding our book, and definitely about your work. You are working amazingly in the field of COVID-19 with saliva. Yes.
Hello, hi, Dr. Gargi, hi, Paul. It's a pleasure to see you all. I, first, congratulations on this wonderful book. I have to really congratulate you, and it's very special to see. For me, I feel very lucky to be friends with you and to contribute with you. And I'm very thankful. Now I have to thank Paul that is here, who introduced me, right, Paul, to so many wonderful people, to Dr. Manuel Jimenez, to Dr. Gargi, and to all these wonderful scientists around the globe, and who is really a great pioneer, and a really a special person when we talk about saliva and saliva diagnostics. So Paul, it's a pleasure to see you and to be with you
It's a
here. And
pleasure to be with you too. I'm glad you could join. It's fantastic that you could join. Yeah. So
thank you.
you have worked with one of the pioneers yourself, Dr. Frank Oppenheimer.
Right.
He's a wonderful man, and I think you studied under him either when you were in Boston, and that's a fantastic piece of your history as well. So, yeah, and you work with
Absolutely.
Dr. Sakira. We're working with Dr. Sakira on a across US, across Canada dental project as well. I think there are 10,000 samples being taken for COVID-19. Walter's somebody special too. So thank you so much for being here, Deborah. It's really nice.
Absolutely. You just mentioned my two mentors, my mentor, Dr. Frank Oppenheim, who is a great pioneer in saliva research. And I feel so lucky to have learned from him. He's a living legend
He
when we
is.
talk about saliva. And it was such an honor to be his student, and then to have met Walter Siqueira, who's now a professor in the University of Saskatchewan, Canada, and also the Dean of Research and Graduate Studies at that university, and also a great pioneer in saliva research. So I feel so lucky to have learned from them, to learn from them every day, and to continue this very important work. And it was very special that Dr. Gargi sent me a copy of the book. So we already have the book here in Brazil. I can tell you, I'm talking to you now directly from São Paulo, Brazil. And it's wonderful to see. And you saw in the beginning when I appeared, I was with masks, because we're still wearing masks in Brazil, both when we're inside and outside the buildings. But when we have no one around, I can take out my mask. That's the rule. So now it's a good time. So what we see here, and Brazil was really, unfortunately, one of the epicenters of the COVID-19 pandemic around the globe. And what we saw is that us that work with saliva and having this expertise in dealing with saliva and working with saliva diagnostics, we saw how we could help to fight this pandemic. So this was very special because we, in collaborative work, so working with Paul, working with Dr. Gargi, working with Walter, with Manuel, we could all work together and to help fight this pandemic and to bring accessibility of diagnostics to the whole world population. And this is only possible through saliva, because this is the cheapest. I mean, you explained this very nicely. This is the most non-invasive body fluid to be collected. Is the cheapest way to collect body fluid. And this gives accessibility. When you come from countries like I do from Brazil and like you do from India, you know the importance of having access to diagnostics. We know this firsthand. So this is what we saw, that us that work with saliva, we could contribute to fight this pandemic. And what we saw is that our scientific discoveries actually helped saving lives. So this was, I feel very blessed to be able to contribute in this fight, and we're still fighting this pandemic. And when we think back, we think all the lives that were saved because they had access to diagnostics, and saliva was a big player in that. So it's very important that you compiled much of the information in the book. And I really, once again, I congratulate you, Dr. Gargi, and I see also Dr. Hujita. After you explained to me how to pronounce the names, I apologize. And your mentor, Dr. Gargi, is here too. We're both blessed to have wonderful mentors, and
Absolutely.
Dr. Paul. So it's really a pleasure to be here and to be able to congratulate on this book today.
Thank you, Deborah. Thank you for all the words of appreciation. Thank you so much. And so the last question, because I think Dr. Paul it's really early there, and I would not hold anybody of you much longer. So, Dr. Paul, I would, as an overview, you can tell us that how has this pandemic, changed the overall disease diagnosis, particularly intimating that saliva may have come of age at last. I mean, it's something like it's a good time for saliva to come up. So we would really like to know from you how you think the disease diagnosis has got a shift, or is it going to shift? Yeah.
So I guess, I've been involved in saliva diagnostics for 25, 26 years, maybe, with 3 different companies. The first 2 companies, pioneers in the area, OraSure Technologies, Saliva Diagnostics. I have to say, we developed an HIV test back in 1996, which was well before its time. The market wasn't ready for it at that time. It took till the year 2000, I think, to actually come up with a test, and it wasn't FDA approved till about 2006. So, it was premature at that time, and maybe that's because the technologies that were available to collect saliva or to be able to test saliva were not quite perfected, and the manufacturing of tools that were available was not that great. And I think probably the most important factor is that nowadays, we all know that analytes that are present in saliva are present in much lower quantities. And so in that respect, you have to have technologies that are much more sensitive to be able to detect salivary biomarkers, maybe proteomics, genomics, transcriptomics. But now we have next generation sequencing, we have mass spectrometry, we have lateral flow that uses fluorescence. Each of these technologies are much more sensitive than technologies that were available maybe 20 years ago. So I think that's been a major factor in making saliva a tool of choice, if you like, for collection purposes. But, the companies like 23andMe and Ancestry.com, who started using saliva back in 2012, 2015 for Ancestry.com, they've now collected millions and millions of specimens from people, and it's become commonplace. It's in the United States, it's commonplace to get your ancestry testing done to find out where you came from. And, when I did mine, I find out that I'm mostly English, Irish, but I have a bunch of Swedish in me as well that I didn't know, those types of things. And everybody, it's kind of a gimmick, but it's a very important role for Saliva that really started to put it on the map. But the thing that really put it to fruition, if you like, is the COVID-19 epidemic, where it's been shown to be a very, very useful specimen. And I just read a couple of articles recently from independent people that are outside the Saliva world, because my views are tainted because I'm involved. Your views are tainted as well. But it was from the head of Roche Diagnostics, or somebody who are not a company that are familiar or involved heavily with diagnostics, and they said, once the COVID-19 epidemic is, it's not going to go away, but once we're in a stage where we're at, saliva is going to be the specimen of choice, because it's become a specimen that everybody relies on right now, and the accuracy is much better than it used to be. So and as I said, Dr. Anne Wiley is a name that you'll probably see a lot in the saliva world, because she published, she first came up with the Saliva Direct test, which is one of the tests that we talk about in the book. But they do millions and millions of specimens every day using the Saliva Direct method, and it's just a simple drool into a cup, or collect with a small collection tool, and then provide the results, by a real-time PCR test, maybe a few hours later. But that's helped to, as you mentioned earlier, it's helped people become much more familiar with saliva and much more comfortable with saliva as well. It's not always a comfortable specimen. I mean, I know, this is not a sexist thing, but ladies don't like to spit. Older people find it difficult to spit. Children don't like to spit either, until they get into the teenage years. So, then it becomes a habit. So, so I think there's a lot of things that have kind of propelled it to where it is, and we just hope that's going to grow, because I'm 66 years. I'm going to retire in the next 5 years, 6 years, but hopefully I'm going to continue the role of trying to promote saliva as the ideal tool, in order for us to be able to diagnose many more diseases that we don't diagnose right now. Maybe rare diseases that Dr. Bill Gates is really interested in, and those types of things. So there's a lot of things that still have to be done. And my statistic that saliva is less than 1% of the world market for diagnostics still prevails. We need that number to be much higher, and then we're going to help people even more. So that's my closing remarks anyway. And I really do thank the whole team here that actually worked on putting this book together. It would not have been possible without Sri Ram, Dr. Rujita, yourself, and I value our friendship, and the fact that I'm able to provide input the way you need it from me. So thank you again.
Thank you so much, Dr. Paul. Thank you so much. I mean, we are all really obliged to you. And I think with that, we will conclude this session. So, Shriram sir, I would request you to give your closing remarks as well. And at the end, I'll just ask the youngest one in the room, that is Dr. Rujita, about dealing all these boring people in this entire project. Yeah, Shriram sir, over
So,
to you.
so just two comments. One is, I think you can actually send this copy of a book to the Indian government agencies, including ICMR, who's actually looking at it, including some of the state governments. I think, Rujita, you should probably look at this possibility, because they wouldn't go to Amazon and buy. But I think you could probably, buy a copy and ship
Yeah,
it to
yeah.
them free of cost. Of course, it may be lying on the table, but don't worry about it, because someone is going to read it. May not be the main officer in charge, but some curious scientist who's going to walk in. It'll be in the library. They may actually start reading at it. So send it to government agencies at your expense if you can. That will be a great thing to do. Number two, the public health centers, especially there are actually community service centers, and there are a lot of philanthropic agencies who are actually doing the diagnostics today. I think you can probably also send it to them. So look at who are all there, but I think you can actually look at them. You can find them in the newspaper or the internet. So this is one part. And the second one is a very kind of a very lighter vein. I would say, Rujita, if you find time, please send the link in terms of who was this Gargi Vachak Navi, the ancient lady scholar who actually compiled all the information, who was very well known in the Vedic period in terms of getting all this data. and she's well known for her debates, for the intellectual knowledge she got from the Vedic perspective. So you can send that link to Paul and many others who may not know that, because it's very important. The name is not a coincidence. There should be something in terms of why Gargi was named as Gargi. So there is a background behind it, and that's about 5,000, 6,000 years of history that actually goes there. So it'll be very curious for them to read, because not just man's ancestry from a physical perspective, but there's also an intellectual ancestry is what I call. So thank you so much for calling me, and thank you, Paul, for staying up early in the morning. Thank you, Ruchita. Thank you.
No problem. Now I need a coffee.
Yes, thank you, Shriram sir. And I think you always you always speak more that the one that I deserve. But I think in the book, the foreword sir has written for this book, Shriram sir has written. I think that is amazing. By seeing that, I feel that okay, so I have to work more hard. I think this is not enough. This is not enough actually. So, Rujita, just to come along, and so how was your experience in this entire project? Because I think you just completed your academic year, and you came inside this organization, and you have been doing fantastic. And I must say to everybody that Rujita has taken up Genomedan, our e-magazine, the free magazine, and she's doing a wonderful job. I think Genomedan is now her. I mean, it's just her space. I don't interfere in that. So,
It's
how
a
was
very good effort, Rujita. It's very good effort, Rujita. Just wanted to appreciate you for your
yes,
efforts.
yes.
Very good.
Thank you, sir. Thank you. Thank you so much. Actually, it's the ma'am's trust and her belief in me and her continuous guidance that I could be able to do this. And I'm very thankful that I've got this opportunity actually. So thank you, ma'am, for everything. And Sriram sir, yes, Vachak Navi Gargi, I'll definitely send the link to everybody. She was at that time, she was the revolutionary, female sage who could bring this. That a girl can become so knowledgetic and such a learned person that she can take up her career instead of a family which was typically meant to be in that era. And she used to have the discussions and knowledgetic arguments with so many great pundits at that time who were all male basically. So definitely I will share the link to everybody. And my experience, I'm the youngest here. I am like, I don't know, like my knowledge and my experience is literally very small. And I'm really privileged to be guided by Dr. Gargi ma'am, Dr. Paul sir's constant, like corrections and everything. How keenly he observes each and every sentence, every word. I have learned a lot. This was a life-changing experience for me. I have learned a lot, sir. Like I have to thank for Gargi ma'am and Paul sir to making me a part of this project actually. And Gargi ma'am keeps telling me all the time, like Sriram sir and Paul sir are the two pillars and two guidance, pillars in her life that she could take a turn. And being in this, amongst these wonderful people, this aura itself gives me a boost to do something different and conventional unconventional in my life. And Dr. Gargi ma'am has been that mentor for me. So she gives me the boost whenever I feel sad. Like I call her up and I like, ma'am, I want to do something different. Let's do something different. And we come up every time Dr. Paul sir, Sriram sir's name is taken in our conversation. We
Yes.
keep you people in front of us and think, if they were in this place, what could they have done? How would they have thought and proceeded if this was the situation given to them? So I'm really delightful to be a part of this. And this book is the first book for me, very special and dearly to my heart. So I'm very thankful, and definitely words fall short for explaining my experience and my happiness with this book.
Thank you, Rujita, and I think you are a very dedicated person. And like I'm sitting in front of my mentors, but then as your mentor, yes, I think you are a very. I think, sir, Rujita has a very keen eye on observation, and she's very dedicated. She's very passionate. So I think,
Very much.
No,
Yeah.
there are 2 points actually. One thing,
Yes,
Rujita,
sir.
that you can learn from Paul is that, as you rightly said, Paul is a diagnostic expert. Most diagnostic experts are very keen observers. So I think good that you said that he
Right.
has corrected you every single turn. So that's one thing. Number two, I think again, as Dr. Paul hinted earlier on, be prepared to have the second version of the book, because a lot of technologies that are emerging between now, especially the Omicron variant, and probably in the next 12 months, are going to be important. So probably in about 12 months, be prepared for the second version, where there's a lot of technological inputs that are going to happen all around in the diagnostics. So just watch out and be prepared. Thank you.
Yep, I agree.
Thank you.
Thank you.
So
Thank you so much. So we conclude this session today, and thank you, Dr. Paul Shriram sir, once again. And I wish that you have a great time ahead. Thank you, Rujita, for being here.
Thank you all
Thank you all.
for
Thank
for
you all.
inviting me here. Have a wonderful day, everybody, and let me know any follow-ups that need to be done on our side.
Yes, yes, Dr. Paul, as always. Thank
Okay.
you.
Thank you
So
so
much.
Thank
much.
you. Thank
Bye
you
bye.
all.
Have a
Bye
good day,
Thank you.
everybody.
Bye
Bye bye.
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 Bye. Bye bye.